Bonani Bonani, Dumelang. Hello everybody, my name is Helen DeWet, and that must mean that we are going to be spending some time together studying life sciences. And of course we would not be here today if it were not for Liberty. Thank you, Liberty, for all you do and have been doing for over 30 years for the education of our young people in Mzansi. You should already have that Tenfold education app on your cell phones and in your back pocket, so right up until the time of the exam you can check out something that you've just forgotten for the moment. You can also follow us on our YouTube channel at Mindset Learn. So we've got you covered, guys, and what we're looking at today is the final revision for November 2025, paper 2 for life sciences. Now, if you were with me for our paper 1 revision, you'll realize that quite a lot of the things that we discussed actually came up in the paper in some way. So I think it's very valuable if you're sitting there together with your note paper, copying down reminders and making sure that you have got the very best final revision in your back pockets. So let's begin, shall we? Paper 2. What are the topics that we will expect to see tomorrow in paper 2? Well, first of all, a large component of the exam will be made up of the DNA section. DNA, the code of life. You're going to need to know about the structure of DNA, the idea and concept and process of DNA replication, and of course DNA in terms of how it is the source for protein synthesis. Meiosis, genetics, evolution, and coupled with that, human evolution. So less topics than you had to cover for paper 1, but hugely important nonetheless. So let's proceed. We're going to start with a lightning revision going through DNA, the code of life. There's a diagram alert here. In some way you are going to have to either draw something or label a diagram relating to the structure of DNA. You need to remember that both DNA and RNA are nucleic acids. They belong to the group of organic molecules known as nucleic acids, and they are polymers, which means that they are made up of building blocks. Now, sometimes those building blocks are structured very simply together, like a string of amino acids making up a polypeptide. But the monomers of nucleotides the monomers of nucleic acids are called nucleotides, and they have a far more complex structure. You must know the structure. At the basis of the nucleotide is the sugar molecule. Now in DNA, you know it is deoxyribose, and it is a pentose sugar. It is bonded to a phosphate molecule, and it is also bonded to a nitrogenous base. In RNA, we're going to see ribose as the sugar, phosphate identical, and some of the nitrogenous bases are the same while others differ. Please remember that your sugar is the molecule, or one of the 3 molecules where 2 bonds are formed, one with the phosphate and one with the nitrogenous base. Important points here when we look at the structure of DNA is that we've got this sugar phosphate. We call that commonly the sugar phosphate backbone. All right, so It's the sides of the molecule. You could call it the backbone. It's also like the two sides of a ladder, and that is hugely important. The rungs across the ladder, or the steps that you would walk on up the ladder, those are the nitrogenous bases, and the nitrogenous base is joined to a complementary nitrogenous base on the other side. And of course, if we've got the two sugar phosphate backbones and we've got our nitrogenous bases on either side, these nitrogenous bases have to be, the word is complementary, not complementary, like, oh, your hair looks very nice today. Not complementary, but complementary, which means that they pair with each other. I suppose something like salt and pepper or eggs and bacon, they go together. And you must remember which are your complementary bases. So adenine and thymine are complementary, and cytosine and guanine. All right, the way you can remember it is there are apples in the tree and there are cars in the garage. We do not find cars in the tree. We do not find apples in the garage. All right, put those two complementary ideas together for your basic structure. And then remember that the phosphate sugar backbone, together with its complementary rungs, forms a double, because there's two of these backbones, a double helix. And the helix means that it is twisted into a spiral shape, and it's got the two backbones. And our nitrogenous bases are held together with hydrogen bonds. All right, moving on now, you may be asked to give us some differences between DNA and RNA. So the similarities are they're both nucleic acids, and they both have the same basic structure, building block of a nucleotide. So if we look at the diagram, we need to read our question very, very carefully. Are you asked to give just any differences between DNA and RNA, or are you asked to give structural differences or functional differences? So the structural differences are what it looks like. So here you would say ribose in RNA, and you would say deoxyribose in deoxyribose in RNA, in DNA, and you would be able to compare them. This is a single strand, this is a double strand. Of course, this has adenine, thymine, cytosine and guanine, but in RNA we have adenine, cytosine and guanine, and we have uracil, which replaces the thymine. So these are all structural differences, what it looks like and how it looks different from each other. When we come to functional differences, we're looking at what are the functions and how do their functions differ. And here we know that DNA carries the genetic code. DNA is responsible for carrying that recipe that makes you, you. It carries the recipe for all of your proteins, for all of your functioning, your enzymes, hormones, everything in your cells. Whereas RNA has a very different function. RNA is going to be involved in protein synthesis. RNA is going to copy that genetic code, and it is going to move into the cytoplasm, and it is going to make the protein. All right, let's move on. This process of DNA replication. Remember that it is not part of mitosis or meiosis. It happens at the end of interphase before cell division takes place. And it is the process during which a DNA molecule makes an exact copy, or, fancy word, a replica of itself. So when we're looking at DNA replication, in our minds we must think of a process that starts with a DNA double-stranded molecule. We see that these, in the second part of the process, these hydrogen bonds break so that the molecules on either side can split apart. When they split apart, they expose empty spaces which are going to be filled with complementary bases that are in the nucleoplasm. We see this as a replication fork. We can see that it's like a road that then splits, a fork in the road. And we call this a replication fork because the DNA molecule is going to split all the way down eventually, and all of these little complementary bases are going to come in to make, to join up and make new complementary base pairs. So when we started with our original ATCG, 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 we have made an identical copy, which is then going to format itself into its double helix. Now, not to confuse DNA replication with the transcription stage of protein synthesis. So this is a common mistake and a common misconception, or a common area of confusion. So don't you fall into this trap. When we're looking at protein synthesis, we know that a copy has to be made of the original gene. Now, in DNA replication, the whole DNA molecule was copied. In protein synthesis, we don't copy the whole DNA strand or the whole chromosome. We only copy the gene that we need to use to make the specific protein. So I want you to think of this DNA is very much like a recipe book, and it's got hundreds and hundreds of different recipes in it, but we only want to make chocolate cake. So we open the recipe book just where the recipe is for chocolate cake, and we can make a copy of just that recipe. And by recipe, we are meaning the gene. So we don't, in protein synthesis, do what DNA replication does, which is copy the entire strand of DNA or the entire chromosome. We just go to the recipe that we want, the gene that we want, and that is copied. And the copy is called messenger RNA. The messenger RNA moves out of the nucleus into the cytoplasm, and it then takes part in the second phase. So this part up here that takes place inside the nucleus is transcription, and that is copying over, right? We're copying over the gene, and inside the cytoplasm we have translation. So translation is changing from our nucleic acid code. It was all in a code made up of nucleotides, and now we are producing a protein. And the protein is a polypeptide, and of course it is using amino acids as its building block. So this translation that takes place is like me saying to you at the beginning of the show, Sanibonani, or hello everyone. That is a translation from isiZulu to English. Now what we're doing here is a cellular translation. We're taking nucleic acid code and we're converting it into amino acid code. Please make sure, this was studied at the beginning of your year, so it might be far in the distant past, and please make sure that you really go through that section and you revise it carefully. Moving on now, we move to the next section, which is really closely related to DNA, is meiosis. You must know the different stages of meiosis, and remember that goes through meiosis 1 and meiosis 2. Now, whether you write 1 or 2, please remember that when you're talking about, for example, anaphase 2, you must use that 2. It's important that you distinguish between the two stages with the 1 and the 2. You must know the purpose or the function of meiosis, and you must also know what happens when meiosis goes wrong. So we need to look at the disorder known as trisomy, which means 3 times the chromosome 21, commonly called Down syndrome. And it is brought about by a non-disjunction. And you must know what a non-disjunction is, that usually chromosomes move to opposite poles in anaphase, but what happens with a non-disjunction is that they do not separate properly, and 3 chromosomes or 4 chromosomes instead of 3 chromosomes, whatever the chromosome number is, but 1 extra chromosome moves to one pole and 1 fewer chromosome moves to the other pole. That is a non-disjunction. And the example that you study in matric is Down syndrome. There are many different examples. Now, this section, if you do not know your diagrams, you're in for a hard time. You must know how to recognize the diagram and the phase or stage of meiosis. You must be able to label everything. You must also know, in terms of the function of or purpose of meiosis, is that yes, it is there to halve the chromosome number to make haploid gametes, but it is also there to bring about genetic variation. This genetic variation takes place in 2 different parts or stages of meiosis. And the first stage are where we see genetic variation being built into the process is during prophase 1. See, I must write which prophase I'm talking about, and it's the process of crossing over. You need to know, and this is what I said about diagram alert, you need to know that during prophase 1, the nuclear membrane, nucleolus, all disintegrate. Spindle fibers are beginning to form, and this is hugely important, homologous chromosomes come to lie close together. And they undergo this process called crossing over or synapsis. So we have the 2 homologous pairs lying together, and they become ambivalent when the 2 chromatids that are lying adjacent or next to each other cross over. This point is known as the chiasma, which simply means X. And when they move apart again, we'll see that from this orange chromosome, we've got a little bit of orange on the blue chromosome and a little bit of blue on the orange chromosome. And this brings about recombination of genetic material. Now that is the first time we see genetic variation built into the process. The second time is during metaphase 1. We must write the little 1 there. The homologous chromosomes now come to lie in a double row at the equator. I like to teach my students metaphase starts with an M, and so does the word middle. So the cells come and line up along the middle plane, and they are each via their own centromere attached to a spindle fiber. Now sometimes the paternal chromosome will be drawn to the top or to this pole, and sometimes, and therefore the maternal one will also go up there. Sometimes it's the maternal one that is at the top and the paternal one that is at the bottom. So this mixing up or randomizing of which of the paternal and the maternal chromosomes moves to one pole, it is completely random. This is known as the rearrangement of homologous chromosomes on the metaphase equator. All right, and that is the second place where we see genetic variation built into the process. You must know and understand that for meiosis, you're not only going to be asked to recognize the diagrams, but you will be asked to draw the diagrams as well. I promise you, you're going to have to draw diagrams. So that sharp pencil, nice, neat label lines drawn with a ruler, all ending in the one plane, all of those rules apply. Lovely large diagrams. You're not paying for the paper, use the paper. Draw those diagrams nice and large. So this is a kind of question where we're not going to draw it today, but we've got to interpret the diagram. We've got to explain, and when you see that word explain, please, matrix, it means use your words. It means use sentences, not just one word. Explain why these diagrams show the same phase, but at different times of meiosis. So first thing we do is, so what is the phase? Look at the diagrams and try and identify what phase are we talking about. And I told you previously, M for middle, and we can see that our chromosomes are lying on the middle of the middle plane of the cell. So M for metaphase. But we've got a slight difference. Here in diagram A, there's just a single row of chromosomes on that metaphase middle equator plate. Here, this is also metaphase, but we've got our double row of homologous pairs. The homologous pairs have not yet split apart. So this is metaphase 1, B is metaphase 1, and A is metaphase 2. You can write that as a 2 or as a Roman numeral. But now it's not just enough to identify that A is metaphase 2 and B is metaphase 1. We have to explain why these diagrams show the same phase, metaphase, but at different times of meiosis. So we would have to use some words and some sentences to say that in metaphase 1, which is shown by diagram B, we see that there is a double row of chromosomes. The homologous pairs have not yet split. But in diagram A, metaphase 2, we see one row of chromosomes, and that means that our number of chromosomes has already halved. And here we're looking at the haploid number, and here we're looking at the diploid number. So we've got to use words. A question like this would probably be out of 2, maybe 3 marks. So make sure that you use your sentences very carefully to explain what is going on. So we've looked at DNA, we've looked at meiosis, and you're going to go home and practice those diagrams. I think we're going to take a short break now, and when we come back, we are going to look very briefly at the third topic in paper 2, which is genetics. Don't go too far away. Make sure that you come back. As soon as we're back, we're going to get on with genetics.
Today's Life Sciences word of the day on the endocrine system is homeostasis. Homeostasis is a process of maintaining a constant internal environment, blood and tissue fluid, within the body. This enables the body to function efficiently despite changes in the external or internal environment.
Did you know that there is a National Sleepyhead Day in Finland? On the 27th of July every year, Finland celebrates National Sleepyhead Day. On this day, the last person found sleeping in any home is woken up by being doused with water by the early risers. The tradition is believed to have started in the Middle Ages, when it was believed that the last person to rise on this day would be lazy for the rest of the year.
Want to catch up on some of your studies? Join Mindset for late night studies. Monday is mathematics, Tuesday is physical sciences, with life sciences on Wednesday and accounting on Thursday. Friday is English, maths literacy on Saturday, and Sunday is a good day to catch up on geography. Every day from 9 p.m. till midnight, and again from midnight till 6 a.m. Late night studies, only on Mindset.
Welcome back, and you're with Helen, and of course that means we are looking at life sciences. Today we're focusing on paper 2. We would not be here if it were not for the generous donations and sponsorship by Liberty. Thank you, Liberty, for what you do for our youth of Mzansi. Of course, you should have me already in your back pocket in your Tenfold education app, but if you haven't yet, please download that. And of course, you can follow us on YouTube channel at Mindset Learn. All right, let's get back to what we were looking at before our little ad break, and we're focusing now on genetics. So, what are the subtopics in genetics that you need to know about? Genetics, remember, is the study of inheritance and variation, how we inherit characteristics via our DNA, but even though we inherit our characteristics from our parents, we show variation. We are not clones of our parents. We've already discussed things like crossing over. We've discussed things about the rearrangement of homologs on the metaphase plate, and all of these perform the function of building variation into the whole process around sexual reproduction. You must know a little bit about Mendel and his pea plant experiments, but more importantly, you need to be able to apply the principles that Mendel hypothesized in monohybrid crosses, and you need to understand how they work in dihybrid crosses as well. Remember, a monohybrid cross means we're looking at one characteristic. A dihybrid cross means we're looking at two characteristics, so maybe eye color and length of hair in guinea pigs or rabbits or hamsters. And in a monohybrid cross, we'd only be looking at one of those characteristics, like eye color, for example. Now, in order to investigate how these crosses work and what the outcome of the crosses are, we use some tools. You will either have been taught how to use the tool a Punnett square, or what I call a drop-down solution. You can use either in the exams. In our marking memos, both options are given, so you can use either, and we're going to look very carefully at those in a minute. You also need to know about pedigree diagrams. When we look at a monohybrid cross and we use the Punnett square method or a drop-down solution, we're looking at one generation. We're saying, here's the parents, and if the parents had certain characteristics, what are the chances that the first filial generation or the offspring will have those characteristics as well? So in those cross diagrams, we're looking at one generation, parent to first filial. We can then convert the first filial into the second parental generation, and we can follow it into a second filial generation. But remember, the Punnett square or the drop-down method only looks at one generation at a time. In pedigree diagrams, we're looking at many generations. We're saying, if this characteristic appeared in the great-grandparents, what are the chances of it appearing in the grandparents, in the parents, and in the offspring? So we're looking at many generations in a pedigree diagram. You also need to know about sex linkage. You need to know the difference between the term autosome, which is a chromosome that carries all of your characteristics, and it's chromosome 1 to 22. Your sex chromosomes, you should have 2 of them. They are also sometimes called gonosomes because they are getting that name from the word gonad, which relates to the word sex. Here we're looking at X and Y. All right, so and this is your next pair of chromosomes. Pairs 1 to 22 are your autosomes, and then the 23rd pair is the gonosome pair. If you have XX, you are a female. If you have XY, typically you're a male. So we need to look at sex linkage, and then if there are errors on any of these chromosomes, let's say this autosome pair is autosome pair number 15, for example, and if there's an error or a mutation on that DNA, what is the result going to be? What is the consequence of that error going to be? And you need also to know what happens if there is a mutation on an X chromosome. What is the genetic disorder. Now, I want to give you a word of warning here. Here I'm going to say, when you're asked that question on genetic disorders, which you will be asked, read the question carefully. If it mentions X chromosome, if it mentions sex-linked, then you must follow the rules for doing an X, let's say it's color blindness with a little C, and this one has got a capital C, so it's got the dominant. You must use X's and Y's. If it does not give this information, you must presume that it is an autosomal genetic disorder, and then you're going to be looking at ideas like capital T, small t times capital T, and doing your normal Punnett squares or drop-down method. Please read that question. If it doesn't say it has a mutation on the X chromosome or that it's a sex-linked disorder, don't presume that it is. And remember, your blood groups are not sex-linked. Okay? Your blood groups are not determined by sex linkage. That appeared in the prelims, and it caused some of my students to make mistakes. So, let's get back to the basic structure of the chromosome and the fact that we have maternal and paternal homologs, always in pairs, and that one comes from the father, the paternal, one comes from the mother. So this one was going to be donated in the sperm cell, and the maternal homolog was in the ovum. And of course this was haploid, and that was haploid, and when we put them together we got the diploid version, that is you. And remember that although there are genes at exactly the same locus for different characteristics, they may be different on each of the homologs, and that's where the word allele comes in. Same gene, it's coding for our blood group, but different allele. Father, blood group A, mother, blood group B, for example. So when we come to this Punnett square, it was named after the man who invented it, the Punnett square. There are certain things you must put into this diagram, and if you don't, you're going to lose marks. And if you draw it all untidy on the page, the marker is not going to go and look for things. Set it out neatly. Use a whole page to set it out if you want to, but set it out neatly. So the Punnett square must show the parental generation. It could be P1, it could be P2. Read the question. And you must always, in the parental generation, you must say what is the phenotype, and you must convert the words. When we look at phenotype, we look at words. Tall, short, dwarf. When we look at genotype, we're looking at a description of our alleles. So capital T, small t, capital T, small t. All of that must be present and correct in order for you to get a mark. Next, we need to show that our marker, that we understand what happens next. What happens next is meiosis. And each of these allele pairs is split by meiosis from 2n to n. This is splitting over here. And we need to write this up for our marker. So it's not just when we'd be playing noughts and crosses here, we are showing our marker that we understand what is happening in this part of the diagram. So you must label it. Remember, a Punnett square is a diagram. So we need to label what we're doing in that diagram. So we write down segregation of alleles into gametes during meiosis. This top row and the first column, label it that is the consequence of meiosis. Then in the body of the table, we are going to perform fertilization. And we're going to show with labels for our marker that we understand what is happening here. That T, that capital T and that capital T are coming together to create a new diploid organism with the allele complement capital T, small t, capital T. And we label this, these are all the possible recombinations of those alleles, all right? And it's fertilization. Here's the one parent, here's the other parent, and now what possibly could the result be? If you have done this correctly, you will have got a mark for that, and you will have got a mark for that. You've got to put the labels in for the mark, as well as get all the components of the diagram correct. You can't just get the mark for labeling or get the mark for filling this in. You've got to show our examiner exactly what is going on. And finally, you get to the conclusion. And this is where you now have to take from the body of the table out into a solution at the end. And we're showing that our second filial generation, because that's what the question told us, is made up of, there we go, 1 capital T, 1, 2, capital T, small t, and 1 small t. That is the genotype. You must then say, I'm now going to convert that genotype into the phenotype, and I'm going to use my words to describe what the phenotype is. Both of those individuals will show the phenotype tall because the T is completely dominant. The capital T is dominant over the small t. And this one will be our little short one. That is the phenotype in physical language, in words. And then finally, you go back and you read the question. What did the question want you to do? Did it ask, for example, what is the percentage chance of being short or having short pea plants? You've then got to answer that question. So the percentage chance of having dwarf or short pea plants is 1 in 3, which is 1 in 4, should I say, sorry, which is 25% chance of having short plants. Read the question and answer it, and there you'll get a mark for that, and you'll get a mark for answering the question. So it's very complicated. But I promise you, remember what you have to do. You have to give both the P1 and F1 genotypes and phenotypes. You've got to show how meiosis happens to produce the gametes. You've got to show fertilization to show the combinations of all gametes. And then you've got to put your answer as a ratio or percentage. Just read the question and see, must I say there's a 1 in 4 chance, or must I say there's a 25% chance? So this is the Punnett square. The drop-down method, you would have done exactly the same. You would have written up your P1 genotype, and we're just making this one up. That's the genotype. You would have written then that this is tall, times tall, that is the phenotype. So that is identical for the drop-down method. Where the drop-down method now varies from the Punnett square is we now put each of those alleles into their own little circle. And that is trying to show meiosis. So label it as such. This is now meiosis, and these are now the gametes. And now I must show the gametes, all possible combinations of those gametes back into the diploid organism. So I use the firsts, insides, outsides, lasts, or FOIL, firsts, outsides, insides, lasts. So we take the first two, and that gives us capital T, capital T. We take the insides, we take those two. We always write the capital first. The outsides, there they are there, little t, and the last. Sorry, the outsides would have been that one, capital T, small t, and then the lasts. And then we've got to, so we've shown fertilization. Now we've got to answer. We've got to say that in the F1, our genotype was, our phenotype was, and then we have to answer the question. So that's the drop-down method versus the Punnett square. You do what you've been taught how to do and what you're comfortable with. You need to remember that there's different kinds of dominance as well. In Mendel's one law of dominance, he said that one allele will always dominate and suppress the other allele. In other words, in the heterozygous condition, capital R, small r, we will only see that dominant genotype being expressed. Codominance is when we have two different alleles that are equally dominant. And so when we put them together, we see, as in this rhododendron plant, we had an allele for pinkness, an allele for whiteness, and it comes together as patches. In animals, we call that the roan. Incomplete dominance is when we put the two genotypes together, the red and the white, for example, we don't get patches, we get a blending together. Please make sure that you do know your different kinds of dominance. And as I mentioned earlier, you need also to be able to understand a pedigree diagram, which shows 1, 2, in this pedigree diagram, 3 generations. You need to understand the key, which will tell you which is the female, which is the male, and you need to be able to work the pedigree diagram to work out whatever the question asks you. Moving on to the very last section of work on evolution. For evolution, you need to be able to discuss theories of origins, fixity, which is linked with ideas about creationism, where all organisms were created in a way and remain identical and unchanged. Lamarckism, who challenged fixity and proposed his notion or his hypothesis, and finally Darwinism. And you need to be able to show how Darwin's understanding of natural selection for how evolution takes place also displaced Lamarckism as a potential theory. You need to be able to demonstrate natural selection as well as artificial selection, and the only difference, it's identical in mechanism, the only difference is that artificial selection serves human goals. We breed animals and plants for our own specific goal in mind. It could be cows that have better milk producing glands so that they are better milk producers. Speciation takes place when a new species is formed from an existing population, and we need to be able to show that evolution isn't something that happened long, long ago at the time of the dinosaurs maybe, but that it is in fact happening today in our own bodies and in the bodies of organisms around us. The basic idea about natural selection is very simple and straightforward. So I'm going to use a little hypothetical example here of some little pink fish and some little blue fish. And we must say that at time 1, when we look at this population, this little population, meaning they're the same species and they're living in the same area at the same time, and there is the potential for breeding. In our population, we have some pink fish and some blue fish, and we see that there's a 1-to-1 allele frequency. It suggests that there's no advantage to being pink or blue, it's just you pink or you blue. But over time, some environmental circumstance changes, and it causes one of the alleles to become more favorable. And by favorable, it means it's conveying a survival advantage. Let's say a flock of birds, a population of birds moves into the area. They can eat fish. They love catching the fish out of the water, but they can see the pink fish in the water easier, and in the water the blue fish are camouflaged. So what happens is we see that over time it has become more of a survival advantage to be blue. And we are going to see that if you're pink, you either get eaten or you get eaten before you can reproduce. You don't pass on your pink alleles. But if you're blue, you've got the survival advantage, and that means that we're going to see over time a change in the allele frequency. Suddenly it's 9-to-1, suggesting that there's an advantage if you are blue. So natural selection of the fitter, and by fitter we don't mean they can swim faster or they're stronger, they go to the gym. We mean they have a better survival advantage over the less fit ones, in this case the little pink ones, drives the process of evolution in this population. And what is the definition of evolution? Here it is. Evolution by natural selection, the change in the allele frequency in the gene pool of a population. And of course it doesn't happen immediately, it happens over time. Darwin hypothesized that the change from one species to another was a gradual event. Eldridge and Gould have said and suggested that no, it follows the model of punctuated equilibrium, that over time we see that there is a particular species, some environmental catastrophe or change takes place, like our bluefish, pinkfish scenario with the birds coming in, and we see that there is change that takes place. And if that change is so dramatic that we can no longer see these new fish as recognizing the old fish and breeding with them, then we would say that there is a new species that has evolved. Punctuated, just like you write a sentence and you punctuate it with marks, we've got species 1, species 2, species 3, we've got equilibrium, and it is punctuated by environmental change that pushes the notion of speciation. So you would have to explain the process of speciation as it is given to you, maybe in a diagram, maybe in a case study like this one, where we've got a river changing its route and therefore separating the population. At the moment, if these bunnies could jump over the river, they could still mate with each other and produce viable offspring. But if they remain in their new environment for long enough, they have to adapt to this new environment. And this process of adapting is exactly like those blue and pink fish. And in adapting, maybe generations later, we introduce the bunnies to each other again, and they don't recognize each other as mates. Look, these little bunnies have developed little spots, so they don't recognize, they don't speak the same bunny language anymore. And that means that we've got a new species. You need to be able to look very carefully at case studies, and case studies are going to be thrown at you and you don't know. You've never heard of this kind of bunny or that kind of deer that lives in Alaska or something. You've not heard of these examples before, but you know the underlying concept of natural selection and speciation, and you apply what you know to these particular new case studies, and you'll be perfect. Go through your process of evolution carefully and be able to work out what all of those steps are. They're given in all of your textbooks, in all of your notes. Make sure that you know them and can apply them in a new situation. But you've got this, you're going to walk into that exam tomorrow morning and you're going to be strong. Survival of the fittest, baby, and you are life sciences fit. Thank you for spending some time with me. Thank you for making sure that you are the best possible class of life sciences students ever. From me, Helen, good luck and goodbye.